The single-stranded DNA is pulled through the aperture of this nanopore. As each one passes through, it creates characteristic modulations in the current that can be used to determine the order of bases on that strand. The workflow for a MinION sequencing experiment is straightforward. Firstly, the MinION is connected to the laptop through the USB 3 cable. The user then launches the Mino software that will run the MinION, and after performing a calibration and quality control procedure, the MinION is ready for the sequencing library. The library is added to a port in the MinION, and the fluid flows across the surface of the sensor chip. After selecting the run protocol and starting the experiment, you can now start to see the raw data produced by the nanopore sequencing the DNA. The nanopore signal is measured by an ASIC in the flow cell and processed by the Mino software. Mino controls the MinION. It carries out several core data tasks and can be used to change experimental workflows or parameters. It provides the user with real-time feedback of key sequencing metrics, such as read lengths and number of events, allowing the user to decide how long to run an experiment. The software runs on a host computer with Mino data files being uploaded to Metricore, an on-demand, cloud-based data analysis and bioinformatics platform. Results of these analyses are then provided back to the user as the experiment progresses. This exchange of data allows real-time sequencing analysis to take place, meaning that experiments can be run to generate the right amount of data to answer the user's question, rather than being controlled by an arbitrary runtime.
On the bioinformatics side, the systems produce full-length, full-read data, whether it's DNA, RNA or protein, in real time. And that means you can treat those data bioinformatically in real time as the machine runs. Now, running bioinformatics in real time has a number of advantages. The obvious one is it reduces the compute cost and the management overhead, but it also means you can look for things in real time. And that means if you have an experiment that's looking for a particular thing, whether it's a significant mutation or the existence of a particular strain in a laboratory swab, you can detect and see that with a required level of confidence in real time. If you can see things in real time, you can then feed that back to the system, and you can move the whole system or part of it onto another sample or another experiment.
Multiple nanopores operate independently across many microwells on the arrayed sensor chip, generating many reads at the same time. Combining this with a continuous exchange of data with a cloud-based analysis system, results can be seen in real time, allowing users to decide when they have enough data to answer their questions.